RFLPs, or restriction fragment length polymorphisms, is one of the kind of older applications of biotechnology. And what this does is it helps us identify genetic variation between individuals. Okay, so I have this picture of these SNPs, these single nucleotide polymorphisms. It's just these mutations that occur at the same site. So again, remember thinking about the sickle cell. That's a difference in the SNP, and we went from it. So people without the sickle cell trait have a T in one particular spot in their genome, and the people with the sickle cell have an A. Okay. So what this restriction fragment length polymorphisms are is ways that we can use those little variations between individuals to get some DNA fingerprinting, or any number of applications. Okay, so this is often used in paternity testing because we expect that parents are going to pass on similar of these single nucleotide polymorphisms, right? So the mutations that your parents got, you're probably going to get. And then we can compare those between lots of different, right? Lots, not just one SNP, but lots of them to get a DNA fingerprinting. So I'll show you, we'll talk about how we do that. Okay, so what we do is we take the DNA from our different samples, right? So from suspected, fathered, and a DNA sample from the child, okay, in these separate tubes. And then we add things called restriction enzymes. And restriction enzymes are going to act like a pair of scissors that are going to read the DNA. And when they get to a specific sequence, in this case, it's GAATCC. I said that wrong, but you know what you can read. Okay. These scissors are going to cut the sugar phosphate backbone at that particular sequence and give us two fragments, right? And of varying sizes. So if we have individuals that have SNPs, right, these little mutations, we're going to get a bunch of different size fragments. Okay. So everyone's going to have a different combination of those SNPs. And that's how we can get this fingerprinting. So again, in the in-person lab where we do the gel electrophoresis, if the suspect had the suspect should give us the same DNA fingerprint as the crime scene. So in this case, so maybe this is our crime scene, and we would say, okay, this person doesn't really match because our crime scene individual, right, the DNA from the crime scene has, yeah, it has these two bands, but the crime scene DNA doesn't have this band. Okay, and so then same thing here, same thing there. But if we look at this one, we're like, oh, this suspect matches what we saw at the crime scene, right? Same thing. This is really kind of silly and funny, but it just shows how cheap and easy this is. My mother-in-law is about to move into, it's a retirement community. It's not a nursing home. It's a retirement community. And one of the rules for her to bring her dog was that they had to get a DNA sample from it so that they could get the dog's DNA fingerprint. Because if there's poop that's not picked up around the complex, they can extract the DNA that is left in the cells that are shed from the dog's gut when it poops, and they can see then whose dog did that poop, and then get whoever was in, whoever left that dog poop, didn't clean it up, to find them or whatever the hell they do. I don't know. But if a retirement community has the ability and the funding to do this, like, it's pretty cheap and easy. So anyway, that's just kind of a fun RFLP anecdote. All right, molecular cloning is the next one. This one's a bigger boy, so I'm going to put it in its own video.